My talk is actually sandwiched between two of the tutorials that Terry's going to be giving you folks. And the reason is that I'm going to be talking about a little bit of an exploratory analysis project that we've been doing here at the Institute. And the idea is to try and integrate some of the data from two different atlases. So specifically the adult mouse brain atlas, which Terry showed you a little bit. I mean, the images weren't working, but hopefully that will be fixed soon. And then also talk a little bit about the mesoscale connectivity atlas, which Terry will be going through in the next session. But the idea is that we'll talk a little bit about the two atlases, and then talk about some analysis methods to link the data between the gene expression atlas and the connectivity atlas, and some interesting findings we can at least start to explore. Finally, I'll spend just a couple of minutes talking a little bit more on sort of some of the analysis that Bosuka talked about with identifying single-cell transcriptomic types, and again, how we can try and start to relate some of that to connectivity-based ideas. So the general goal when we started this project, which is in some sense a little bit of a side project, it's not tied specifically to any of our main goals, but it's a little bit more on the exploratory side. But the goal was to look to see if we can find relationships between gene expression in the adult mouse brain and mesoscale connectivity. This is not a new idea. There's been previous work that has looked at this type of analysis, most notably Hongwei Dong in 2009, who looked specifically at the hippocampus to identify domains that were related in terms of connectivity and also in terms of gene expression. In 2011, Leon French published a paper that looked at our gene expression atlas. At that point, we didn't have the connectivity atlas, so he actually took rat connectome data and did some analyses to tie that together. More recently, there's been a couple of papers that have looked at the voxel level data from both the gene expression and the connectivity atlas, and then tried to relate those two to build a predictive model. And the reason that all of this is possible, so this is again a slide that Jane showed, in 2004 we came out with the adult mouse brain atlas, which looks at 20,000 genes in the sagittal plane and then 4,000 of those also sectioned in the coronal plane. But the reason we can do these analyses across these different atlases is actually because they are all put into the common coordinate framework. So you can register these brains, as Terry pointed out, and then you can actually look at a specific region of the brain in gene expression space, and then look at that same region in the brain registered to the same common coordinate framework in the connectivity atlas. So you actually have a powerful way using basically the API. You can pull everything down from the API or use the SDK. You can get gene expression data for multiple genes, 20,000 genes. You can get connectivity data, all of which is in the same coordinate framework. And so that allows you to do some very nice comparisons or integrations among these two data modalities. This is just a slide showing the connectivity atlas. Terry's going to talk about this in more detail, but the general idea is that we have stereotaxic or functionally targeted injections to over 300 brain regions throughout the adult mouse brain. We actually can visualize the connections at the mesoscale level from those injection sites all over the brain, as you can see in this series of images. And then, again, everything can be put together informatically into a common framework, so you can look to see across all these injections which sources are actually projecting, or cells in which injection sites are projecting out to what targets. So all of this is actually, the details about how this is done has been published in a paper by Sungwook Oh and Julie Harris from Hongwei Sung's group. And so all the details are there. They're also on the website if you have questions about the experimental methods or the way the informatics platform is designed. So what we wanted to do is first to do a very gross measure about to examine how gene expression and connectivity could be related. So on the informatics level, we already have everything registered into the same reference space. So we don't have to worry about re-registering some of the data or to try and have some sort of mapping between, okay, we've called this voxel in the gene expression space belongs to this structure, what is the corresponding structure in the connectivity? All of that is done for us already through the atlases, and all of that is accessible, which makes all of this downstream analysis very simple. So what we wanted to do is first to take gene expression, gene by gene, and to develop essentially a co-expression matrix. So the idea is we take gene expression over all the regions. So what you saw in the demonstration was a little histogram that covered various coarse regions and quantified the gene expression in each one. We now have a much finer, or what we're using here is a much finer demarcation of the regions. There's about 210 that we're calling. We z-score the expression for each gene across all the regions, and then we actually just take the outer product of this which gives us this region-by-region co-expression. It's a very coarse measure of the relatedness of two different regions in gene expression space based on a single gene. So we generate one of these matrices for every single gene that we're working on. So then what we do is we take the connectivity matrices. So what we've done is when we look at all the injection sites in the Conductivity Atlas, the experiment doesn't the injections don't behave automatically in the sense that they don't always confine themselves to a single region. They don't always incorporate the entire anatomical region. So what was published in the paper was a linear model of connection weights, which is essentially deconvolving all of the connectivity data such that you actually have region-to-region specified weights. So instead of injection to source, we've now converted it to source region and target region. So that's also downloadable from the paper as a flat file, so you can get that very easily. So once we have this connectivity matrix, which is essentially a large square matrix, 200 regions on one side, 200 columns on the other side, and a series of weights that connect each of the regions to each of the other ones, we can actually look at three flavors of this type of matrix. We can look at the weights directly. We can also look at the correlation of two regions as source. In other words, not necessarily how strong this blue and this red region are connected to each other, but how much they overlap in their projection patterns outward to other regions. You can also look at correlation as target, which is, again, if we look at the blue and the red region, how strong is there similarity of patterns of projections that are coming inward to that region? So if you think about it for a little more strongly, if you're talking about gene expression and relating it to connectivity, to some of you it may be clear intuitively which of these three would at least suggest the best relationship in terms of gene expression and connectivity. To others, maybe it's not that clear, but it will be clear in the next slide. Sorry, the slide after this. So the idea is we have these gene expression matrices, co-expression matrices, for every gene. So we have 20,000 of these that were derived from the Adult Brain Atlas. We have a connectivity matrix. We don't have 20,000 of these because these are not gene-based. These are actually just three flavors: the weights, the correlation as source, and the correlation of target. And we apply a very crude metric. We just take the correlation over these two matrices, we shuffle them to obtain a p-value. So we get for each gene, how strongly does its global expression pattern or co-expression pattern correlate with the connectivity pattern across the entire brain that we get from the mesoscale level. We've been looking at the entire brain. Those of you who are interested, you can download the data, look specifically at subcortical regions. You can look specifically across cortical regions, across cortical layers, any subset of the regions that you want. You can always download the data from the gene expression and the connectivity matrices and perform the same analysis. So if you look at the set of 20,000 genes and their correlation values, so each one gets a correlation value to the connectivity matrix, and then you just plot a histogram of the correlations of all of these genes and their correlation values, what you can see is that the correlation values are significantly higher when you compare the genes to the correlation as source. When you compare them to the weights, there are very few genes that actually show significant correlation with the overall connectivity matrix. When you look at them as target, you see a little bit more of a shift towards the right, so you get a few more significant genes. But the most strong signal comes when you look at the correlation as source target. So this, if you think about it, this may or may not make sense. If two regions are connected to each other, that doesn't necessarily mean that they have the same gene expression. However, if two regions have the same projection pattern, then you might think that if there are genes responsible for a specific type of projection pattern, that those genes would be highly co-expressed in those regions. And so that's the hypothesis that we're forming from our original sort of preliminary analysis, that it's actually the overall projection patterns, not necessarily the direct strength of connection between regions, that is most defined by adult gene expression. And if you actually take these highly significant genes from the as source, you clearly can't read this, but you can trust me, I'm going to read off the top categories, go categories, when you run a standard gene expression enrichment analysis. And you get genes that belong to neuron projection, cell projection, axon, and then a whole bunch of other things. So this is a good verification that the genes that we're picking up as being highly correlated using our method are indeed responsible or have been reported in the literature to be related to neuronal connectivity. So the caveat here, of course, is that all of this data is from the adult mouse. So a lot of connectivity is laid out developmentally, so all we're picking up is some signature in the adult gene expression that's still relevant to connectivity. So this could be maintenance of projection patterns. This could also just be residual signatures from connectivity that has been laid down in development. We don't have a developmental connectome. We do have a developmental gene expression atlas, but we don't have a developmental connectome. But that would actually be very interesting to follow up to see which of these genes are also responsible in development for establishing these connections. So the next step is, okay, we know that there's some correlation, at least, between the gene expression when you look at as source or as target correlations and the connectivity matrix. So the next step was, okay, let's take some of these very highly correlated genes and let's just see if we can actually predict gene expression based on, predict connectivity based on the gene expression. So ultimately the idea is that you give me a region, if we can train a model on all of our data, you give me a region and its gene expression, I can predict with some level of success which regions that specific region of the brain will be connected to. So this is great because we don't have, there's some regions in the brain for which we have very low coverage in terms of our connectivity, but we can try to artificially fill out or at least predict the connectivity of those regions that are more sparsely sampled. We use a very standard generalized linear model to do this. The cross-validation step is we actually leave out one entire source region. So we train the model on 212 regions and we say, all right, tell me what the connectivity is going to be of the region that we've left out. And we actually use a two-step process. We first train the model only on the gene expression, not allowing for interactions among genes. We identify which of the 50 genes are most strongly correlated to it just in this very non-interactive model using a standard regularization process. And then we derive a new model that takes the interaction terms from these 50 genes. There's many different ways you can do this. We just picked a very simple one to see how well we could do. So if you take as your starting set 200 genes with high correlation with the connectivity, that's from our previous analysis, and we look at the actual connectivity weights that are derived from our linear model from the Conductivity Atlas, and then the predicted values that we've done that we get from our model, you see that there is some degree of correlation. It's not perfect. There's a little bit of scatter in this data, so it's a good starting point. As a control, if you just pick 200 random genes, you don't get very good prediction of the connectivity. So not surprisingly, the genes that are highly correlated with connectivity do allow you to generate a model that has much better prediction power than if you just take 200 random genes. So we're actually continuing with this analysis. We're developing a new, more refined linear model for connectivity that includes a much more high-resolution parcellation of the brain. And then we can also look more carefully at some of these regularization constraints to try and narrow down some of this, the spread in this correlation. We're also looking specifically, if we take it the other way, instead of starting with just genes with high correlation with connectivity, what if we take functionally defined genes? So categories like cadherins or ephrins, how well do those do at being able to predict the connectivity? So all of this is happening. Perhaps after the next hour when Terry shows you the Conductivity Atlas, you can go home and you can do this and finish this project before we do. That's perfectly fine. So for the last portion of this, I just want to talk a little bit about trying to go into the single cell level and look at cell types and connectivity. This is going to be all informatics, and as you'll see, it's a very broad, in some sense, crude analysis. What is happening, and what was actually published in the paper that Bosilka talked about, is an experimental way to connect cell types and connectivity using retrograde labeling and then profiling cells that actually have specific connectivity patterns at the single cell level. So this will continue at the Institute on the experimental side, which will be a very, very nice data set linking these two modalities. But at the moment, we can actually take our single cell data set that Bosilka talked about and that you can explore on the vignette, and we can say, okay, instead of using all the genes, suppose we take a functionally defined category of genes, like ion channel genes, and just repeat the clustering using only those 400 or so ion channel genes. So what I'm showing you here is these are all the various inhibitory or GABAergic types that we found in our study using the full transcriptome data. And when you do the same clustering using just the subset of 400 ion channel genes, you get the segregation that's much coarser, but it actually makes sense. So you get your NDNF cells here, all your VIP interneurons here, you get your somatostatin groups combining into one cluster except for one that's very, very distinct. And then your parvalbumin types in the cortex, you get two broad clusters when you just use the ion channel genes for clustering. If you use, as a control, if you use 400 random genes without any functional characterization, you might be able to separate essentially your CGE-derived interneurons from your MGE-derived interneurons. But 400 genes picked randomly doesn't give you much power to segregate these types. So it's not surprising that ion channel genes, because they're related to function, should be able to distinguish these cells, especially because we already know, and if you go back to the cell types database, you can see that the firing patterns of these broad classes of cells is actually dramatically different, even at the intrinsic level. If you do the same thing using now 400 axon guidance or cell surface genes, these are a proxy for genes related to connectivity, you get a very similar segregation of your broad cell types. You don't get a segregation of the parvalbumin cells. You do get one of these somatostatin types coming off additionally as its own. But in general, the degree of resolution is about the same among these two. Now, when you go to the excitatory neurons, which are known to have long-range projections and receive input from multiple regions of the brain, when you use just the ion channels, you get a broad separation into your superficial or layer 2 groups. Your layer 4 and your layer 5A seem to be very similar in terms of their gene expression from the ion channel level. And then in layer 5B, you do get two groups that are separate. And interestingly, in layer 6, you get a separation between layer 6A and layer 6B. Again, you get your broad layers that are separating out. There's enough of a signal in your gene expression, in just your ion channel gene expression, to separate these out. When you look more specifically at connectivity, you actually get much better segregation of your excitatory types, unlike in the inhibitory case where there was not much of a difference. So you get your layer 2s, your layer 4s, 5A is now its own group, 5B is its own group, and there's some additional types that split off. And perhaps most interestingly, you now get segregation between your cortico-thalamic and your cortico-cortical layer 6 types, which you don't see when you look just at the ion channel genes. So the idea is that for the excitatory neurons, it appears that including some of these connectivity genes actually allows you to further refine the classification as compared to using just the ion channel genes or 400 random genes. And in some sense, this is tied to what I was talking about previously, in that the best relationship in terms of gene expression seems to be when you compare to a connectivity that's actually relating cell types by who they project to, so the correlation as source. And we know that for the excitatory neurons, especially in the layer 5 and 6, that they have multiple different projection targets, so it's not surprising in some sense that we see better segregation. So just to finish up, we can actually take the global and the cell type data and then start to correlate it with some of the connectivity patterns that you can also get from the Conductivity Atlas, as Terry will show you in a little bit. And then you can use the gene expression to predict connectivity weights in a generative model if you pick the right starting set of genes. And then you can also look at the single cell level and get some general ideas that connectivity-related genes might be segregating some of your projection neurons better than if you use ion channel genes or transcription factors, which is another category we've been looking at. So to learn more about how to access this underlying data for all of this, especially for those of you who are informaticians or like to do data analysis, getting all this data is actually very easy, and then parsing it and playing with it is actually where the fun happens. So Terry will talk to you a little bit more about the Conductivity Atlas and how you can use that data to explore connectivity relationships throughout the brain. We've seen this slide multiple times, but again, we have to thank Paul and Jody Allen for their generous support, and then the work of all of these people to put together these atlases and then to make the data accessible. So for example, I was not involved at all in the Conductivity Atlas or the Adult Mouse Brain Atlas, but I was able to just take the data and do some of these analyses. So, thank you.